To explain the phenomenon of heredity, biological information must be accurately copied and transmitted from each cell to all of its progeny. 3 ways for DNA molecules to replicate can be considered, each obeying the rules of complementary base pairing. These 3 possible ways in which DNA can replicate I will illustrate by having this black be the old strand of DNA and this blue being the new. Okay. The first model of DNA replication would be conservative replication. So if this is your DNA parent strand, then conservative replication has the daughter strands being 1, the intact old, and the other being 2 strands of newly replicated DNA. This is conservative replication, our first possible method of DNA replication. Our second, dispersive replication, would occur with the parent strand mixing and matching with the daughter strand in a mismatched pattern. And both daughter strands would get an uneven amount of both new and old strands, like this. And that's our second possible method of DNA replication, and that's called dispersive again. Now the last possible method of model for DNA replication is semi-conservative. And in this instance, each daughter strand has 1 template strand of DNA and 1 newly replicated DNA, like so. And this is the third model of DNA replication, number 3. So, just to reiterate, conservative replication would leave intact the original DNA molecule and generate a completely new molecule. Dispersive replication would produce 2 DNA molecules with sections of both old and new DNA interspersed across each strand. Semi-conservative replication would produce molecules with both old and new DNA, but each molecule would be composed of 1 old strand and 1 new strand. Now, to figure out which model is which, you would have to perform an experiment. And now 2 scientists, Mendelson and Stahl, were well acquainted with these 3 models, and they decided that they would do an experiment to find out which model was correct. So, they reasoned that if there was a way to distinguish old versus new DNA, it should be possible to test each model and see which one was right. So the scientists decided that they would use a technique called cesium chloride gradient differentiation to decide which replication model was correct. And they were able to determine that DNA replication is semi-conservative, and each strand acts as a template for the synthesis of a new DNA molecule by the sequential addition of complementary base pairs. thereby generating a new DNA strand that is the complementary sequence to the parental DNA. Each daughter molecule ends up with one of the original strands and one newly synthesized strand, like so. So, in conclusion, the correct model of DNA replication is our third model, semi-conservative.